For decades, scientists believed that circadian rhythms, the internal 24-hour clocks regulating biological processes, were exclusive to complex multicellular organisms. This fundamental assumption has been dramatically overturned in one of microbiology's most profound paradigm shifts. Join us as we explore how the discovery of bacterial timekeeping mechanisms has transformed our understanding of life itself, challenging our concepts of biological complexity, evolution, and even the nature of awareness. From molecular clockworks to philosophical implications, this is the story of how the simplest organisms on Earth revealed one of biology's most sophisticated systems. Our story begins in 1986, when Takao Kondo's laboratory in Japan detected something that would eventually upend established scientific dogma. They observed what appeared to be circadian rhythms in cyanobacteria, specifically in Synechococcus elongatus. At the time, this finding was so contrary to accepted knowledge that it was initially dismissed as experimental error. The scientific community remained skeptical. How could single-celled organisms without nuclei possibly maintain sophisticated timekeeping? The controversy persisted for nearly 2 decades until 2005, when researchers achieved what many thought impossible. They successfully reconstituted the complete cyanobacterial circadian oscillator in a test tube using just 3 proteins, KaiA, KaiB, and KaiC. This minimalist system could maintain precise 24-hour timing without requiring gene expression, a capability previously thought exclusive to complex organisms with intricate regulatory networks. What makes the bacterial circadian system truly remarkable is its elegant simplicity and astonishing precision. Unlike the transcription-translation feedback loops that drive timekeeping in plants, animals, and fungi, the cyanobacterial clock functions through cyclical phosphorylation and dephosphorylation of KaiC proteins. The breakthrough came when researchers demonstrated that these isolated KaiC proteins could maintain a precise 24-hour oscillation for days when provided with ATP as an energy source, the first successful in vitro reconstitution of circadian systems. Subsequent structural studies revealed that KaiC forms a hexameric ring that undergoes conformational changes throughout the day-night cycle. This molecular cogwheel effectively measures the passage of time through protein-protein interactions and phosphorylation states. Perhaps most impressively, the cyanobacterial clock demonstrates temperature compensation, maintaining consistent timing across various environmental temperatures. This remarkable precision allows cyanobacteria to anticipate daily changes in light conditions, optimizing photosynthetic machinery hours before sunrise and protecting sensitive processes from damaging UV radiation during peak daylight hours. The true paradigm shift came in 2019 when researchers at the University of California, San Diego, led by Dr. Susan Golden, discovered functional circadian rhythms in non-photosynthetic bacteria. Their team identified a minimal circadian oscillator in Bacillus subtilis that regulates metabolic activities and stress responses with 24-hour periodicity. This discovery shattered the assumption that bacterial timekeeping was limited to photosynthetic organisms that needed to track sunlight. Even more surprising was the 2021 discovery by the Puka laboratory at the Netherlands Cancer Institute. They found that certain soil bacteria possess temperature-compensated timekeeping mechanisms that function across diverse environmental conditions, a defining feature of true circadian systems. These findings triggered a wave of investigations across diverse microbial phyla. By 2022, researchers at the Max Planck Institute for Terrestrial Microbiology identified rhythmic gene expression patterns in anaerobic methanogens from deep-sea habitats, suggesting that even bacteria evolving in environments without direct sunlight exposure maintain temporal organization. The molecular mechanisms varied considerably between bacterial lineages. Some utilized post-translational modifications similar to cyanobacteria, while others employed novel RNA-based oscillators or metabolic cycles tied to cellular redox states. As research expanded, scientists discovered an astonishing diversity of bacterial timekeeping systems. The human gut bacterium Bacteroides thetoetamicron possesses a 24-hour rhythm that synchronizes with host feeding patterns through bile acid sensing pathways. Meanwhile, environmental isolates of Pseudomonas fluorescens demonstrate seasonal rather than daily rhythms, with molecular oscillators tuned to longer time scales that track environmental changes across months rather than hours. This diversity suggests that bacterial timekeeping has evolved multiple times to address different ecological challenges. Some bacteria maintain daily rhythms tied to light-dark cycles, while others track host behaviors, seasonal changes, or even tidal patterns in marine environments. The mechanisms are equally diverse, from protein phosphorylation cycles to metabolic oscillators and RNA-based timing systems. This remarkable variety of solutions to the problem of temporal organization demonstrates the evolutionary importance of timekeeping across bacterial domains. The discovery of widespread bacterial timekeeping has profound evolutionary implications. Molecular clock analyses now suggest that rudimentary timekeeping mechanisms likely evolved over 3 billion years ago in the earliest bacterial lineages, predating eukaryotic circadian systems by over 1 billion years. Dr. Martha Merrow at Ludwig Maximilian University of Munich proposes that circadian timekeeping may represent one of life's most ancient adaptations, potentially as fundamental as DNA replication or energy metabolism. This challenges our understanding of evolutionary complexity and suggests that temporal organization is a foundational property of life itself. Comparative genomic studies published in Nature Microbiology in 2023 identified homologous timekeeping components across diverse bacterial phyla, suggesting a common ancestral origin for many bacterial clock systems. However, the substantial diversity in molecular mechanisms also points to multiple independent evolutionary inventions of timekeeping, representing a striking example of convergent evolution. This suggests that temporal organization provides such significant fitness advantages that it has been repeatedly selected for throughout evolutionary history. The discovery also offers new perspectives on the endosymbiotic events that led to mitochondria and chloroplasts. Recent evidence suggests that the circadian systems of these organelles may have evolved from bacterial predecessors, with vestiges of bacterial timekeeping mechanisms still functioning within eukaryotic cells, creating a layered temporal architecture within complex organisms. The bacterial chronobiology revolution extends far beyond theoretical importance into practical applications. Researchers at ETH Zurich have engineered bacterial clock systems as biological sensors for environmental monitoring. These bacterial chronometers can detect subtle environmental changes and maintain timing records without requiring continuous observation. By designing bacteria with circadian outputs tied to specific environmental triggers, scientists have created living sensors that can monitor pollution levels, temperature fluctuations, or chemical contaminants over extended periods. Unlike traditional sensors that require constant power and data collection, these bacterial systems can autonomously record temporal patterns of exposure and maintain this information until retrieval. This approach has proven particularly valuable in remote environmental monitoring, where traditional electronic sensors would be impractical. The bacterial timekeeping mechanisms provide a biological memory of environmental conditions that can be read days or even weeks after exposure events. In medicine, the discovery of bacterial timekeeping has opened new avenues for understanding host-microbiome interactions. Research from the Weizmann Institute of Science has demonstrated that gut bacteria maintain their circadian rhythms distinct from their hosts, influencing metabolic processes, immune function, and potentially even neurological health. Clinical studies now suggest that disruptions to bacterial timekeeping through shift work, jet lag, or irregular eating patterns may contribute to metabolic disorders and inflammatory conditions. A 2023 study in Cell Metabolism demonstrated that time-restricted feeding protocols can resynchronize gut bacterial rhythms in patients with metabolic syndrome, improving glucose tolerance and reducing inflammatory markers. This research suggests that maintaining proper temporal organization within our microbiome may be as important to health as the species composition itself. The timing of bacterial activities, from metabolite production to immune system interactions, appears critically important for maintaining host microbiome balance. These findings are reshaping approaches to conditions ranging from irritable bowel syndrome to diabetes, suggesting that chronobiological interventions targeting bacterial rhythms could complement traditional treatments. The insights from bacterial chronobiology are transforming agricultural practices as well. Researchers have developed chronobiologically optimized probiotics for livestock that synchronize with animal feeding cycles, enhancing nutrient absorption and reducing the need for antibiotics. By ensuring that beneficial bacteria are metabolically active at precisely the times when feed moves through the digestive system, these timed probiotics maximize fermentation efficiency and nutrient extraction. Similarly, soil inoculants with engineered bacterial clocks have shown promise in improving plant microbe interactions by aligning microbial activity with diurnal patterns of root exudation. Plants release different compounds from their roots throughout the day-night cycle, and bacteria with synchronized timekeeping can optimize their metabolic activities to match these patterns. This chronobiological approach to agriculture represents a shift from simply considering which bacterial species are present to understanding when they are active, a temporal dimension to microbial ecology that promises more sustainable agricultural practices with reduced chemical inputs. The pharmaceutical industry has begun exploring bacterial clock mechanisms as novel antibiotic targets. By disrupting pathogen timing systems, researchers hope to develop antimicrobials that reduce virulence without creating the selective pressure that leads to resistance. Early preclinical studies with KaiC protein inhibitors have shown promising results against cyanobacterial blooms without harming other aquatic organisms. This chronotherapeutic approach represents a fundamentally different strategy in the fight against bacterial pathogens. Rather than attempting to kill bacteria outright, which inevitably selects for resistance, these clock-targeting compounds aim to disrupt the temporal coordination of virulence factors. Many pathogenic bacteria rely on precisely timed expression of virulence genes, coordinating invasion strategies and toxin production. By scrambling these temporal programs, new antimicrobials could render pathogens less effective without directly threatening their survival, potentially reducing the selection pressure that drives resistance evolution. This approach could be particularly valuable against biofilm-forming bacteria, which use temporal coordination to build their protective structures. Perhaps most profoundly, these discoveries challenge our philosophical understanding of consciousness and awareness. If even the simplest life forms possess mechanisms to anticipate environmental changes and organize their existence temporally, what does this suggest about the nature of biological awareness? As chronobiologist Dr. Carl Johnson of Vanderbilt University noted in a 2022 perspective piece, the discovery of bacterial circadian systems forces us to reconsider what we mean by awareness. These single-celled organisms, without neurons or sensory organs, nevertheless maintain an internal representation of time that allows them to anticipate and prepare for regular environmental changes. This realization has sparked interdisciplinary dialogues between microbiologists, philosophers of mind, and cognitive scientists. Some theorists now propose that anticipatory behavior, the ability to prepare for future conditions rather than merely react to present ones, may represent a primordial form of consciousness that emerged early in life's history. In this view, the bacterial clock represents not merely a biochemical oscillator but a rudimentary form of temporal cognition that allows organisms to exist in relationship with time itself. This perspective challenges traditional boundaries between reactive biochemistry and cognitive processes, suggesting that the foundations of awareness may extend far deeper into the tree of life than previously imagined. As we look to the future, bacterial chronobiology stands at the frontier of multiple scientific disciplines. Researchers are now exploring how bacterial timekeeping might function in extreme environments, from deep-sea hydrothermal vents to the human bloodstream during fever. The discovery of temperature-compensated clocks in non-photosynthetic bacteria raises intriguing questions about what other environmental cycles bacteria might track. Could there be bacterial timekeeping systems synchronized to geomagnetic fluctuations, atmospheric pressure changes, or even host hormonal cycles? Synthetic biologists are working to harness bacterial clock mechanisms as modular components for engineered biological systems. These could range from time drug delivery systems to environmental remediation bacteria programmed to activate specific metabolic pathways at predetermined intervals. Meanwhile, systems biologists are developing comprehensive models of how bacterial timekeeping integrates with other cellular processes, potentially revealing fundamental organizing principles of life. As our understanding deepens, bacterial chronobiology promises to continue challenging our assumptions about biological complexity while offering new tools for medicine, agriculture, and environmental science. The bacterial chronobiology revolution represents one of the most significant paradigm shifts in our understanding of life. What began as a controversial observation in cyanobacteria has expanded into a fundamental reassessment of biological complexity and the nature of temporal organization in living systems. We now recognize that the ability to structure existence in time, to anticipate rather than merely react, may be one of life's most ancient and fundamental innovations. The discovery that bacteria keep time challenges us to recognize that temporal organization is not a luxury of complex organisms but a core feature of life itself. From the molecular clockworks of the KaiB proteins to the diverse timekeeping systems found across bacterial phyla, these mechanisms reveal an unexpected sophistication in the simplest life forms. The implications extend from evolutionary biology to medicine, agriculture, and even philosophy, forcing us to reconsider what we mean by biological awareness and the relationship between life and time. As research continues to unravel the molecular mechanisms and ecological significance of bacterial chronobiology, we find ourselves at the beginning of a new chapter in our understanding of life's fundamental properties. The timekeepers within the simplest organisms may yet reveal profound insights about the nature of life itself and our place within time's continuous flow. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.